the chapter on cell reproduction. The topic for today is just going to be the structure of DNA when it comes to cell reproduction. I don't know if you even remember way back to probably chapter 4, I would guess, when we talked about cell organelles and the form of DNA in the nucleus. When a cell is not dividing, the DNA is in the form of a molecule called chromatin with an IN. This is the stringy form of DNA. So imagine, I think the analogy that's used to give you an example of how long DNA is, they say if you imagine the nucleus, just the nucleus is the size of a basketball, the DNA would be 64 kilometers long. So imagine if you have a basketball-sized ball of string that's 64 kilometers long, and you have to copy that so that you've got two 64-kilometer long strings, and you have to make sure that these strands of string get separated into two new basketballs and have exactly the same copy of information on each side. That's kind of a tedious task. It might not be difficult, but it's certainly complicated and long. And so what happens is before reproduction takes place, the chromatin is going to condense and coil into structures called chromosomes. And I have a really good animation of just how much wrapping and coiling and twisting and condensing chromatin goes through to form chromosomes. So imagine if you take your 64 kilometers of string and you chop it up into, let's say, 46 sections, and each section is scrunched into a little knot. Okay, so basically, instead of having 64 kilometers of string, you have 46 little knots of string, and then that's much easier to copy those 46 knots and then separate them so that you make sure that each new basketball has exactly the same 46 knots as the original did. So that's going to be the topic for today. At the bottom of the screen, you see we're going to go backwards. So we start with the DNA double helix. You, in a cell at any one time, you never, I wouldn't say never, you almost never see the actual double helix. You will only see the double helix in that form when DNA is being copied or when proteins are going to be made. In a couple of chapters, we go over protein synthesis and how you go from copying the code on DNA to actually making proteins. So if you need to access the information on DNA, which is in these little like green and blue and red and yellow blobs that you see there, because my picture is so blurry, that is when you will see the double helix. 99% of the time, the DNA that you see in the nucleus is in the form of chromatin, and what you're seeing right here, this is chromatin. It's actually DNA wrapped around proteins. I'm going to talk about that in a second. Then you're going to have this is going to, you see how it's like twisted around and condensed into this structure, which doesn't really have a name. And then this is actually going to wrap around like the old-fashioned telephone cords. And then this is actually going to be part of a chromosome. So again, I've got a video that will kind of show you the animation of how that's done. So we've got 3 billion pairs of nucleotides in DNA, and that is what contains all of the information that makes each of us an individual. Okay, so I already spent time talking about, 64 kilometers. All right, here we go. So I'm actually going to run this backwards.
Individual units.
Shush. Because I find that we all recognize the DNA double helix, and I would rather go backwards instead of starting with chromosomes, because we all have grown up with visualizing what the double helix looks like. So again, the double helix is going to wrap around these the spherical shapes that you see here, these are proteins that are called histones. Histones are literally just the proteins that DNA wraps around so that it helps to maintain the shape of the chromosome. So DNA wrapped around histones, that is chromatin. So you see this is going to twist and fold into this structure. And then this is going to coil around like a telephone cord. And then this is going to be one of many strands that you see here in this bunch, which has taken out a chunk of chromosomes. So if you go from a chromosome, you zoom in, you see like all of this thready material. Those threads are made of coils, and the coils are made of threads of chromatin, and that's chromatin. And then the little tiny green line that you see, that's the double helix. Okay, so that's how you take a 64-kilometer strand of string and you scrunch it down into chromosomes. Chromosomes are easy to separate. So if you know that you've separated your DNA into different segments and each segment condenses into a chromosome, then you can take the chromosomes and separate them easily. So the form of DNA during cell division is called a chromosome. And like I said, histones are the proteins that the DNA double helix wraps around. This helps to maintain the shape of the chromosome, that very characteristic X shape that we use to depict or to picture a chromosome. We also have proteins called non-histones. These are involved in controlling the activity of specific regions of DNA. You have to remember that DNA contains all of the information that makes each of us a human being, also individual, but it also contains a whole lot of extraneous information that's never, ever, ever accessed. If you compare the DNA molecule between a human and a chimpanzee, I do believe that a chimpanzee is 99.8, if I remember correctly, 99.8% identical to a human DNA molecule. That means the only difference in all of the 3 billion nucleotides in a human being is 0.2% that separates us from being chimpanzees. This is why they are our closest living relative. If you looked at living things as being a family, chimpanzees would basically be our, I guess, brother or sister, because our DNA is not exactly the same, but we come from the same quote unquote parent or ancestor. Okay, so here you've got, oh, I'm actually wrong. I identified this as being chromatin. I should have identified the step after that as being a chromatin fiber. So that's my mistake. So you got the DNA double helix. We wrap around histones. And this is called a nucleosome. I forgot that step. And then finally, nucleosomes are going to condense, and you see how in the middle, the fiber kind of, the DNA kind of folds in on itself. That's chromatin. So it's even more condensed than the double helix. Like I said, the only time that you find DNA in this form is if you actually have to access the information on the nucleotide bases, which are in the middle, that form the steps of the ladder. Otherwise, when it's in this form, you cannot access that information. So a lot of our DNA is permanently in this form, just because, like I said, I think it turned out that about 80% of our DNA is just not used. Okay, I'll say that again. 80% of our DNA is not used ever, like ever in our entire life. It's just there. So imagine what kinds of genes, genetic information you can find in those sections that are never accessed. A lot of our DNA, which you'll learn in a couple of chapters, is just nonsense. And so you might think, well, Teacher Renee always explained that in biology, natural selection, or like the universe, whatever, the process of living always favors more efficiency. So there's always a purpose to everything. So if there are sections of DNA that don't code for anything, it just seems to be nonsense that's kind of shoved in there. There has to be a purpose for that or a reason for that. And over the course of the next couple of years, those are things that I'll be talking about.
In this animation, we'll see the remark.
Okay, what I want to point out first, what makes this totally awesome is that we now have the computer technology and the electron microscope technology that we can actually create computer models. Oh, sorry. Ka, yuban ka. Ka. Kapun ka. Sorry, I have cleaner being delivered. What was I saying? Right. So we have the technology to be able to understand exactly what these molecules look like. So we can actually use computer generated models to show us what it actually looks like. Right. So this isn't like out of somebody's imagination, like, oh, this is what I imagine DNA looks like. What you're looking at here, that's what DNA actually looks like. If we actually had the power to zoom in and look at a DNA molecule in three-dimensional form, this is what it would look like. This is why I love these animations.
We'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. Next, the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin.
So this is the form that DNA takes when the cell is not dividing. Chromatin.
This fiber is then looped and coiled yet again. Leading finally to the familiar shapes known as chromosomes, which can be seen in the nucleus of dividing cells.
So this
chromosomes are not
video.
always present. They form around the time cells divide when the 2 copies of the cell's DNA need to be separated.
Okay, so that is how DNA coils to form chromosomes. Now, we have to look at the structure of chromosomes. So we notice that each chromosome is made of 2 identical halves. Now, these identical halves are because DNA is copied before division. Now, the reason why DNA is copied is because the whole point of cell division is to create 2 brand new identical cells. So therefore, you have to copy the DNA so that each new cell has exactly 1 copy of the entire DNA molecule. So each half is called a chromatid, not to be confused with chromatin, which is the stringy form of DNA. So we say they are sister chromatids. They contain exactly the same information, okay? It's important to remember that, exactly the same information. So when you divide, you're basically going to say this is 1 chromatid, that's the other chromatid. And when the cell divides, you're going to split those 2 chromatids apart so that each new offspring cell has exactly 1 copy of that chromosome. Now, the constricted area in the center is called the centromere. So sister chromatids are attached at the centromere. What you're looking at in this picture is a photograph of chromatin. So you see that it's not as tightly condensed as chromosomes, but it is still pretty loopy, I guess. It would be difficult to try to read information on these strings without actually uncoiling it further. So that's called chromatin, right? That's what DNA looks like most of the time. Now we have to remember the difference between prokaryotic cells and eukaryotic cells. Prokaryotes do not have a nucleus, so their DNA is floating freely inside the cytoplasm, usually in an area called the nucleoid. And remember, oid means similar to. So it's like a kind of a nucleus. It's similar to a nucleus, but it's not a nucleus. You also notice that you have two kinds of DNA in a prokaryote. You have the chromosomal DNA, which contains the information to create, oops, sorry, that's my chair, contains information to create new bacteria, but it also has, in addition to that, something called plasmid DNA. This is going to come back in when I start to talk about DNA technology and how we can copy genes. A lot of times we use plasmids in bacteria to help us produce materials from genes. How do you call it? Like insulin is a great example. Insulin used to be made, I think, using cows or pigs, I can't remember, but it was really expensive, and obviously you have to rely on other living things. And then when we developed gene technology, we were able to actually take the gene, like first we were able to identify the gene that codes for the information to make insulin, which is a protein. And then we're able to cut that gene out of human DNA, and we were actually able to splice it or attach it to plasmid DNA inside of a bacteria. So now we literally use bacteria to grow insulin for us. Pretty cool, but that's a whole other chapter. Right. So the prokaryotic DNA is much simpler, obviously, than eukaryotic DNA. It's a much smaller molecule. And you can see based on the diagram that it's also circular. It's not stringy like eukaryotic DNA is. It's a single loop. And it might contain one or maybe two chromosomes. Something that I'll talk about in a second is that the number of eukaryotic chromosomes is not dependent on the complexity of the species. I'll talk about that in a minute, but here's a video explaining why bacteria reproduce so quickly because of the size of their DNA.
Prokaryotic cells are much smaller and simpler than eukaryotic cells. It takes prokaryotic cells about 20 minutes to divide. Eukaryotic cells take much longer to prepare for cell division. Eukaryotic cells have to copy more DNA and build new organelles. It takes eukaryotic cells about 24 hours to divide. A single bacterium can turn into 1 million bacteria in about 6 hours. After 24 hours, a single bacterium will become about 2,360 billion bacteria, while a single eukaryotic cell will divide just once to produce 2 cells.
So 2 reasons why prokaryotes can divide so quickly. One, their DNA is much smaller. Actually, 3 reasons. Not only is their DNA smaller, but it's also in a loop. So if you imagine you've got a single loop, all you have to do is you've got replication starts in one direction and goes in the other direction, and then you meet in the middle. And at the end, you've got two DNA molecules. A eukaryotic cell, the DNA is much more complicated. It's a lot bigger. And the third reason is that prokaryotic cells don't have organelles. So it doesn't have to worry about having to make additional mitochondria, Golgi, ER, all that kind of stuff. So that's why it can reproduce every 20 minutes. Okay, so this is what I was going to bring up before, or what I did bring up before, but I wanted to talk about. Each species has its own characteristic number of chromosomes. For human, that number is 46. For the orangutan, which is here, that's also a close cousin of ours, it's 48. This is the main reason why two different species cannot interbreed. A human and an orangutan cannot have offspring because we have different numbers of chromosomes. If you have two species that are super closely related, for example, a horse and a donkey, they might have, I'd have to actually look it up if they have the same number of chromosomes. Nature in general doesn't like two different species to interbreed. And so having different numbers of chromosomes is one way to prevent that. If you have a human, their egg and sperm is going to have 23 chromosomes each, because egg is going to be 23, sperm 23, because then when we reproduce sexually, the egg and sperm fuse together, forming a set of 46, which is a human. So for an orangutan, they're going to have 24 each. So if you have a human and an orangutan that mate, you're going to have 23 plus 24, which is 47. 47 chromosomes is neither a human nor is it an orangutan. That doesn't mean that two different species have, two different species have managed to reproduce successfully. The most common example I can think of is a horse and a donkey. They breed together to form what's called a mule. However, nature kind of takes care of the fact that a mule exists by making the mule sterile. So mules cannot reproduce on their own. Like, they can't reproduce at all. So there's two ways to describe whether or not you can have kids. One is fertile, that means you can, and the other one is sterile, which means you cannot. And sterility can be due to many different reasons, whether it's a lack of sperm, the sperm isn't healthy enough to fertilize an egg, or maybe the eggs aren't released on time or at all, things like that. There's a whole plethora of reasons for that. So you have two different species. You've got horse and donkey, and together they make a mule, but the mule is sterile. So that's nature's way of being like, okay, you got away with it this time, but like, I'm not going to allow a mule to reproduce. Another example is tigers and lions. I know that it's either a tigon or a liger, depending on which one is female, which one's male, but I can't remember which one's which. But in general, this is one way that nature has of preventing interspecies breeding. The other thing that I wanted to point out was having a look at the number of chromosomes. So you've got Penicillium, which is a prokaryote. They have between 1 and 4 chromosomes. You have Saccharomyces, which is a yeast, has 16 chromosomes. A mosquito, you would think that a mosquito, being an insect, which is quite high up there on the evolutionary scale of complexity, would have a lot more chromosomes than a yeast cell, but it doesn't. Then you've got a housefly, garden pea corn. Check out the adder's tongue fern. This is a fern. Ferns are prehistoric plants. They've been around since when the dinosaurs were roaming around, and they can have between 480 and 1,000 chromosomes. So this just gives you, hopefully gives you the idea that just because an organism is more complex, for example, humans, we like to believe that we are the masters of the universe because we have technology, we can burn down the planet. We certainly seem like we're trying our best to ruin the planet at the moment. That doesn't mean that we are genetically complex. We only have 46 chromosomes compared to other species that have, I mean, look at the dog even has 78, almost twice as much. So there's no relationship between the number of chromosomes and the complexity of the organism. Okay, couple of last things before we finish today. We can separate the set of chromosomes into two groups. So first we have the sex chromosomes. You can see them pictured here. This is taken with a scanning electron microscope. How do I know? Because it kind of looks 3D. And so you see the X chromosome and the Y chromosome. Please take note that the Y chromosome is much smaller than the X chromosome. This means that there are many genes that are on the X, which you cannot find on the Y chromosome. So if there is a mutation, and baldness, like losing your hair, is a really good example of this. Baldness is carried on the X chromosome. So if, for example, your mother's, like for the guys in the class, if your mother's father is going bald or is bald, then probably your mom is carrying the gene for baldness. And if that X chromosome is the X chromosome of your XY pair, and your Y chromosome doesn't have any other alternative form, you're going to go bald. So it's always good, if you're worried about going bald, look at your grandpa on your mom's side, look at your mom's dad, and that will tell you what you need to know. So this is why there are some genetic abnormalities that affect only men and not women, because women have two X chromosomes. So if we do have some kind of abnormality or mutation on one of the X chromosomes, we have another X chromosome that might have the normal version of that gene, and we won't show any symptoms or any characteristics of that illness or that condition. So these two chromosomes by themselves, they are the 23rd pair, because we look at our chromosomes as 23 pairs. Each pair represents one from your mom and one from your dad. So in this case, this would be your maternal chromosome, maternal meaning from your mom, and this would be the paternal chromosome, paternal meaning from your dad. Now, for a long time, if you like history, like I don't like learning history, but I really love watching TV series that take place in history, like Bridgerton and the Tudors and what was the other one? I don't know, anything that takes place in like not medieval England, but like back when the kings and queens and all that. There's tremendous pressure on the women in royal families to produce male heirs, because of course women cannot become king or queen. You can't have a queen, you have to have a king, because apparently, you know, throughout history, we live in a patriarchal society. Again, patriarch, there's that patra again, that means men, right? Or males. So they didn't know anything about chromosomes, and they always assumed that because women were the ones that got pregnant, it's the women's decision, or it's the women's fault whether or not they have a male or a female. Now that we understand how genetics works, we understand that women, that's women, are XX and men are XY. So the female only ever has an X to donate to her offspring. There's no options for anything else. So if humans are going to create male children, then the sperm that has to fuse with the egg has to be a Y sperm. So interesting that there were many queens, especially during Henry VIII. He's the guy that had Queen Elizabeth I. I love that period of time. But he married 8 different women during his lifetime. And he even changed the national church so that he could get divorced, because I think they were Catholic before and you can't get divorced in Catholicism. So he was like, okay, I'm just going to change the church into the Church of England. And so it's like a mixture of Protestant and Catholic, something like that, if I remember my history right. Just so that he get a divorce, because he didn't want to be married to, I think, what was her name? Catherine of Spain anymore. And then when he got tired of his wives, he just had their heads chopped off. Nice guy. Anyway, so these are sex chromosomes, that being the point. Everything else are called autosomes. So here you see what's called a karyotype. Karyotype. It's basically just a picture or a diagram of a set of chromosomes. Like I said, we have 23 pairs. So you see all 22, and then this is pair number 23, which are the sex chromosomes. Again, you see the difference in size between X and Y. Now, if you look at one pair of chromosomes, one of them will be from mom, one of them will be from dad. And that's why we say they're 23 pairs. Okay, so together, these are called homologous chromosomes, right? This is a homologous pair. They don't necessarily contain exactly the same genetic information, but they contain the same type of information. What I mean by that is, let's say this is chromosome number 1. And on chromosome number 1, you find the gene for, let's say, whether you're right or left-handed. That gene is going to be in the same location on each of these homologs or homologous chromosomes. Now, let's say mom is left-handed, dad is right-handed. So on your mom's, on the chromosome that your mom donated, it's, what did I say? She was left-handed. So she has the gene for handedness, but the version on your mom's chromosome that she gave you is left-handed, on your dad's is right-handed. We'll get into which one takes over and which hand you end up writing with in the next chapter when we talk about genetics, but these are homologous chromosomes. They have the same genes, but I want to distinguish different versions. Did I spell it right? Different? I don't know. That looks weird. Is it different? Oh my gosh. Do you ever have that when you like, all of a sudden you look at a word, you're like, wait, is it spelled right? Whatever. Different versions. Okay. Because they don't necessarily have the same information. I want to make that clear. Right? And then this is a karyotype, again, a photograph of chromosomes. So just to drive the point home as far as what homologous pairs mean. So you have this is a chromosome pair. These are homologs. So this would be like pair number 1. When your cells get ready to reproduce, I should probably make that separate. Okay, chromosome number 1, you have 2 different versions. When your cell gets ready to divide, each of these homologs will be copied. And so you're going to have sister chromatids of your paternal chromosome and sister chromatids of your maternal chromosome. And these are the ones that will get separated, into 2 new cells. Okay, so hopefully that makes it a bit more clear. And the last thing is the difference between if you have 1 set of chromosomes or 2 sets of chromosomes. Most of our cells, in fact, every single cell in our body except for females, are egg cells, and males, your sperm cells, they are called diploid, di meaning 2. They have 2 sets of chromosomes, so 2 times 23. We've got 46, all 46 chromosomes, 2 copies of each chromosome. So you have both homologous chromosomes, all of your cells except sperm and egg. All right, we abbreviate this as 2N. Okay, you can see that up here, 2N. Now, for the egg and sperm cells, they have only half of the set of chromosomes. These are called haploid, or 1N. Now, the reason why these are haploid is to ensure that when sexual reproduction takes place later in life and egg and sperm fuse together, you get a full set of chromosomes for the organism. For humans, okay, again, is 46 chromosomes.